Lecture CLS Microbiology Bacterial Pathogenesis. Objectives: list and devise steps of bacterial pathogenesis from its entry to the appearance of the disease. Discuss the importance virulence factor of the infecting bacteria. List and explain the host reaction against invading bacteria. Pathogenesis of bacterial infection. Introduction. The pathogenesis of bacterial infection include initiation of infectious process and the mechanism that lead to the development of signs and symptoms of the disease. Characteristics of bacteria that are pathogens include transmissibility, adherence to the host cells, invasion of host cells and tissue toxicity, and ability to evade the host defense mechanism of the host immune system. Many infections caused by bacteria that are commonly considered to be pathogens are inapparent or asymptomatic. Disease occurs if the bacteria or immunological reaction to their presence cause sufficient harm to the person. Bacterial pathogenesis, or how microorganisms or bacteria cause disease. The first step, entry to the host. The bacteria must access or enter to the host, after that adhere to the host tissue, and then penetrate or evade host defense mechanism. The last step is damaged tissue to cause disease. So we're gonna talk about the first step, entry to the host. What are the portals of entry? The portals of entry, there are three main portals of entry are mucous membrane, skin, parenteral route. There are three main portals of entry: mucous membrane, skin, and parenteral route. The first portal of entry is mucous membrane. Mucous membrane is the epithelial cell lining the respiratory tract, gastrointestinal tract, genitourinary tract, conjunctiva. Respiratory tract is the earliest and the most frequently used entry site for some types of the microorganism. Microorganism or microbes inhaled into the mouth or nose in droplet of moisture or dust particle. For example, common cold, pneumonia that caused by bacteria, which is Streptococcus pneumoniae that cause pneumonia, and there is another type of bacteria also cause pneumonia. And tuberculosis that caused by bacteria, Mycobacterium tuberculosis that cause TB اللي هو مرض السل. Gastrointestinal tract. Another common entry site for another groups of bacteria. Enter through the water, food, contaminated fingers and fomites, must resist stomach SCL and enzyme and bile, if for example salmonellosis. Gastro and genitourinary tract, entry site for most sexually transmitted disease pathogen. For example, gonorrhea and syphilis. Gonorrhea caused by bacteria, which is Neisseria gonorrhoeae that cause gonorrhea. Syphilis caused by bacteria, which is Treponema pallidum that cause syphilis, which is sexually transmitted disease. Conjunctiva, membrane covering eyes and eyelid, for example, trachoma, which caused by bacteria, which is its name is Chlamydia trachomatis that cause trachoma. The entry site must the bacteria must enter from the membrane covering eyes and eyelids that cause conjunctivitis. The second portal of entry is through the skin. Unbroken skin is impenetrable for the most microbe. Some microbe can access through the wound, hair follicles, and sweat gland. Some can bore through the intact skin. Some other produce enzymes that break down keratin. Keratin, it is important protein in the epidermis. Its function to adhere cells to each other and to form a protective layer on the outside of the skin. So some microorganisms can produce enzyme and cause a breakdown to the keratin, which is an important protein in the epidermis. The third portal of entry is parenteral route. Microbes are deposited directly into the tissue beneath the skin or mucous membrane. For example, microorganism يقدر يدخل عبر injections, bites, cuts, wounds, surgery, punctures and splitting due to swelling or drying. ومن المايكرو يدخل عبر واحدة من هاي الروتس راح يسبب infection. Now we have preferred portal of entry. Many microorganisms have a preferred portal of entry which is a prerequisite to cause disease. يعني المايكرو كل مايكرو عنده مكان مفضل يدخل من عنده ويسبب الانفكشن. إذا دخل من غير مكان ما يسبب any infection. For example, Streptococcus pneumoniae that are inhaled can cause pneumonia. يعني إذا استنشقناها ممكن تسبب pneumonia. If swallowed, generally don't cause any disease. إذا بلعناها ما تسبب أي disease. فكل microorganism عنده preferred portal of entry. من يدخل من عنده يلا يسبب الانفكشن. إذا يدخل من غير route ما يسبب أي infection. Number of invading microbes. Higher number of pathogens increase the likelihood of developing disease. كل ما زاد عدد الباثوجينز داخل الجسم ممكن يزيد الفرصة إنه يسبب disease أو infection. عندنا two terms، واحد LD50 and ID50. LD50 من اسمها LD معناتها lethal dose for 50% of host number of microbes that will kill 50% of inoculated test animals. ID50 infectious dose for 50% of the host number of microbes that will cause demonstrable infection in 50% of inoculated test animals. اللي هو infectious dose الكمية من المايكرو أورجانيزم اللي تدخل بالجسم حتى تسبب الانفكشن. For example, infectious dose 50 for Vibrio cholera is 10 power 8 cells اللي هو 100 مليون cells. إذا دخل هذا العدد للجسم ممكن يسبب الانفكشن، لكن إذا دخلت للجسم كمية أقل من هذا العدد ممكن ما يسبب infection. As we say, in order to cause infection, the bacteria must enter to the host and then adherence to the host. Adherence attachment between microbe to the host cell that requires adhesion أو ligand اللي هو روابط اللي هي الموجودة على المايكروأورجانيزم، الريسبتورز المستقبلات الموجودة على الهوست tissue. Adhesion or ligand surface molecule on the pathogen that bind specifically to the host cell surface molecules may be located on a glycocalyx, fimbriae, viral capsid or other surface structure. Glycocalyx. Glycocalyx is the glycoprotein polysaccharide covering the cell membrane of some bacteria. Fimbriae. What is fimbriae? Fimbriae is the protein appendages that found on many gram-negative and some gram-positive bacteria. It used to adhere the bacteria to each other and to adherence to the host cells. Viral capsid. Viral capsid is a protein shell of the virus. So this is the adhesions اللي هي روابط الموجودة على البكتيريا. The receptors, surface molecule on the host tissue to which pathogen adhesion site bind اللي هي المستقبلات اللي ترتبط الروابط من البكتيريا أو المايكرو أورجانيزم ترتبط بالمستقبلات الموجودة على الهوست اللي هي هاي يسموها adherence اللي هي الخطوة الثانية أو second step to cause infection. هذا المخطط يوضح شرحنا how microorganism cause disease. The microorganism must enter the host مثل ما شرحنا عبر mucous membrane, skin, parenchymal root. Second step, the microorganism must adhere to the host. شرحناها how the microorganism adhere to the host. لازم يكون number of microorganism ضمن infectious dose 50 حتى يسبب infection. اللي هنا شرحنا هسه راح نكمل بالسلايدات القادمة. The microorganism must penetrating or evasion of host defense mechanism. لازم microorganism يكون عنده capsule or cell wall component or enzymes حتى يسوي penetrating. The last step, the microorganism cause damage to the host cell. Either direct damage or microorganism produce toxins such as exotoxins and endotoxins or endotoxins or hypersensitivity reaction. بعد ما دخل المايكرو أورجانيزم والتصق بالهوست سوى adherence بالهوست how bacteria penetrate the host defense mechanism. أما البكتيريا يكون عندها كبسول أو محاطة بكبسول يسموها capsulated bacteria أو عندها cell wall component or enzymes تساعدها على penetration. So the first one is capsulated bacteria. If the bacteria is capsulated, so it increases the virulence of it or of many pathogens. Example: Streptococcus pneumonia تعتبر capsulated bacteria, Klebsiella pneumoniae أيضا, Haemophilus influenza, Bacillus anthracis and Yersinia pestis. كل ما كانت البكتيريا capsulated يعني virulence of the bacteria يكون أكثر. Resist host defense mechanism by impairing phagocytosis. من البكتيريا عندها كبسول راح تسوي impairing للفاغوسايتوسيس من تدخل للجسم أو تمنع الفاغوسايتوسيس. Host can produce antibodies to the capsule which attach to the microbe and allow phagocytosis. بعد فترة ممكن الهوست سيل يفرز antibody ضد الكبسول ويساعد على الفاغوسايتوسيس. لكن بالبداية طبعا البكتيريا اللي عندها كبسول راح تسبب الانفكشن لأنه الكبسوليتد بكتيريا تساعد إنه تسوي evade للهوست ديفنس ميكانيزم. As we say how bacteria penetrate host defense mechanism. أما تكون محاطة بكبسول أو capsulated bacteria أو عندها cell wall components أو تفرز إنزيمات. So cell wall components. أما تكون البكتيريا عندها أما protein or waxes. أما protein found on the cell surface and fimbri of Streptococcus pyogenes mediate attachment and helps resist phagocytosis. AMA protein is a virulence factor produced by species of Streptococcus, which is Streptococcus pyogenes, and it is anti-phagocytosis. Waxes. Cell wall of Mycobacterium tuberculosis contain of wax or consist of thick layer of wax that help resist digestion after phagocytosis and can multiply inside the white PC. Microbial enzyme or extracellular enzymes or exoenzyme that lyse cells, form or dissolve clot and dissolve material in the tissue. ما هي أنواع الإنزيمات اللي تفرزها البكتيريا؟ leucocidin, hemolysin, coagulase, bacterial kinase, hyaluronidase and others. Leucocidin destroy white blood cells that are phagocytes produced by Staphylococci and Streptococci. يفرز الليوكوسيدين إنزيم يسوي دستروي للوايت بي سي اللي تسوي فاغوسايتوسيس فتمنع الفاغوسايتوسيس. هيمولايسين destroy red blood cells or RBC produced by Clostridium perfringens that cause gangrene and Streptococci. Coagulase produce clot in the blood which may wall off site of infection from the immune response produced by some Staphylococci اللي هو Staphylococcus aureus اللي تفرز إنزيم الكواغوليز. البكتيريا Staphylococcus aureus من تدخل للجسم وتفرز إنزيم الكواغوليز راح يحول السوليوبل فايبرينوجين الموجود ببلازما الدم إلى insoluble فايبرين. هذا الانسوليوبل فايبرين حيترسب على جدار البكتيريا الديبوزيت on bacterial cell wall وتجعل البكتيريا مقاومة للفاغوسايتوسيس. Bacterial kinase break down a clot produced by body to isolate infection made by streptococci and staphylococci. Hyaluronidase enzyme break down hyaluronic acid which holds cells together in the connective tissue made by some streptococci and gangrene causing clostridium فإذا تسبب الغاز gangrene بسبب إفراز البكتيريال إنزيم hyaluronidase enzyme. نكمل أنواع الإنزيمات اللي تفرزها بعض أنواع البكتيريا. الكولاجينيز break down collagen which forms connective tissue of muscles, skin and other organs produced by several clostridia. Necrotizing factor causes necrosis to the tissue or kill body cells. Hypothermic factor decrease body temperature. Lysogenase enzyme that destroy plasma membrane of the cell. Protease enzyme that break down. How the bacteria penetrate to the host cell. ممكن بعض أنواع البكتيريا تفرز invasions or cadre. Invasions surface protein that alter actin filaments of host cell cytoskeleton, allowing microbe to enter the cells. For example, Salmonella typhimurium and Escherichia coli that secrete that proteins. Cadre. It is a glycoprotein that bridge junction between cells, allowing microbe to move from one cell to another. After the bacteria penetrating the host cells, how bacterial cells damage the host cells? By three mechanisms: either direct damage, or the bacteria produce toxins or hypersensitivity reactions. Note, most bacterial damage is carried out by toxins. Direct damage. Some bacteria can induce cells to engulf them, for example, Escherichia coli, Shigella, Salmonella, and Neisseria gonorrhea. Microbial metabolism and multiplication kills the host cells. Other microbes enter the cells by excreting enzyme or through their own motility. يعني البكتيريا تكون موتايل. فعن طريق واحدة من هاي الستبس the bacteria can cause direct damage to the host cells. Bacterial cell can damage host cells either by direct damage or toxin production. Toxin. What is toxin? Toxin is poisonous substances produced by the microbe. Frequently, toxins are the main pathogenic factor or the main virulence factor. Toxigenicity is the ability of the microbe to produce toxins. Toxemia. The presence of toxin in the blood called toxemia. Toxin effect may include fever, cardiovascular problems, diarrhea, shock, destruction of RBC and blood vessels, and nervous system disruption. Of 220 non-bacterial toxins, 40% damage eukaryotic cell membrane. There are two types of toxins, exotoxins and endotoxins. What are the differences between exotoxins and endotoxins? Exotoxins are produced inside mostly gram-positive bacteria and some of gram-negative bacteria as a part of their growth and metabolism. They are then released into the surrounding media. والبكتيريا ما تعاني lysis رأسا هي تفرز الإكزوتوكسين into the surrounding environment. While endotoxins are a part of the outer portion of the cell wall, which is called lipid A, of gram-negative bacteria. Only gram-negative bacteria. They are liberated when the bacteria die and lysis. من البكتيريا تعاني lysis and the cell wall break apart, so the endotoxin produced or released into the surrounding environment. Exotoxins. Exotoxins is a protein or enzyme that carry out a specific reaction, while endotoxin is lipid or lipopolysaccharide. Exotoxin is soluble in body fluids, rapidly transported throughout body in blood or lymph. Exotoxin produced mainly by gram-positive bacteria and some of gram-negative bacteria. Most genes for exotoxins are carried on plasmid or fates. What is a plasmid? Plasmid is a small DNA molecule within the cell that is separated from chromosomal DNA and can replicate independently. It carries genes, for example, antibiotic resistant genes or genes for toxins. Phages. What is phages? Phage is the virus that infect and replicate within the bacteria. It composed of a protein that encapsulated a DNA or RNA genome. So, the most genes of toxins are carried either in plasmid on plasmid or phages. هاي للاطلاع تعريف البلازميد والفيتس. Produce inside exotoxin produce inside bacteria and release into the host tissue without lysis of the bacteria. بعكس الاندوتوكسين لازم بكتيريا تعاني lysis يلا يخرج الاندوتوكسين. Responsible exotoxin responsible for disease, symptoms and or death. There are three types of exotoxins: cytotoxins, neurotoxins, and enterotoxins. Cytotoxins kill or damage host cells. Neurotoxin interfere with nerve impulses. Enterotoxin that affect lining of the gastrointestinal tract. Antibody called antitoxin provide immunity. Antitoxin provide immunity. Toxoid. What is toxoid? Toxins that have been altered. Toxoid. Toxoid is a toxin that have been altered by heat or chemicals used as a vaccines for diphtheria and tetanus. Now we have important exotoxins or examples on exotoxins. Diphtheria toxins, erythrogenic toxin, botulinum toxin, tetanus toxins, Vibrio enterotoxins, and Staphylococci enterotoxins. Diphtheria toxins, Corynebacterium diphtheria when infected by a phage carrying toxin. هذا النوع من إكزوتوكسين هو cytotoxin that inhibit protein synthesis in eukaryotic cells. This cytotoxin have two peptides, A active and B binding. Erythrogenic toxin, Streptococcus pyogenes produce three cytotoxins which damage blood capillaries causing a red rash. Botulinum toxin produced by Clostridium botulinum. هذا النوع من التوكسين هو neurotoxin that causing flaccid paralysis. Tetanus toxin produced by Clostridium tetani. هذا النوع من التوكسين هو neurotoxin that cause tetanus. Vibrio enterotoxin produced by Vibrio cholerae. It has two polypeptides, A active and B binding. The A subunit of enterotoxin causes epithelial cells to discharge large amount of fluids and electrolytes. So we can see the rise water stool of cholera patient. Staphylococci enterotoxin, Staphylococcus aureus produce an enterotoxin similar to cholera toxin and cause Staphylococcus food poisoning. Other enterotoxin produced by Staphylococcus aureus cause toxic shock syndrome. Endotoxin is a part of outer membrane surrounding gram-negative bacteria. Endotoxin is lipid portion of lipopolysaccharide called lipid A. Effect excreted when gram-negative cells die and cell wall undergo lysis and liberate endotoxins. while بحالة الإكزوتوكسينز لا السيلز تبقى نفسها وتفرز الإكزوتوكسين إلى surrounding environment without lysis. All produce the same signs and symptoms. أهم الأعراض اللي يسببها الإندوتوكسين هو الفيفر, chills, weakness, general aches, blood clotting and tissue death, shock, and even death. Can also induce miscarriage. Fever is the pyrogenic response is caused by endotoxins. أهم أعراض الإندوتوكسينز هي الفيفر هي pyrogenic effect, cause pyrogenic effect. Another differences between exotoxins and endotoxin. Endotoxin do not promote the formation of effective antibody, while in case of exotoxin, antibody called antitoxin provide immunity. So the conversion of exotoxin to toxoid is possible, while the conversion of exotoxin to toxoid is not of endotoxin to toxoid is not possible. Organisms that produce endotoxin include Salmonella typhi, Proteus species, Pseudomonas species, Neisseria species. While in case of exotoxin, what is the organism that produces exotoxin? For example, Staphylococci, Vibrio cholera and others اللي شرحناها بالسلايد السابق. Medical equipment that has been sterilized may still contain endotoxin. Why medical equipment that has been sterilized may still contain endotoxin? Because endotoxin is stable in 250 degrees centigrade, heat stable, while exotoxin is heat liable. In 60 to 80 degrees centigrade we can نقدر نتخلص من الإكزوتوكسين. We have Lumulus amoebocyte assay LAL is a test used to detect tiny amount of endotoxins. هذا يستخدموه التست على مود يبحثون بالميديكال equipment عن الاندوتوكسين. هل هي موجودة فيها اندوتوكسين أو مو موجودة؟ Events leading to fever or endotoxin and fever response. Gram-negative bacteria are digested by the phagocytes. Lipopolysaccharide is released by the digestion in vacuoles, causing macrophage to release interleukin-1. Interleukin-1 is carried by blood to hypothalamus, which control body temperature. Interleukin-1 induces hypothalamus to release prostaglandin, which reset the body thermostat to a higher temperature. المخطط بالسلايد اللي بعده راح يوضح لنا هذا الكلام أكثر. Endotoxin and pyrogenic fever response or fever response. A macrophage ingests gram-negative bacteria. The bacteria is degraded in the vacuole or lyses in the vacuole, releasing endotoxin. من البكتيريا تعني lysing راح يخرج endotoxin that induce the macrophage to produce interleukin-1. Interleukin-1 is released by the macrophage into the bloodstream through which it travels to the hypothalamus of the brain. Interleukin-1 induces the hypothalamus to produce prostaglandin, which resets the body thermostat to a higher temperature, producing fever. So this is the event that leading to fever in case of endotoxin. Shock, any life-threatening loss of blood pressure. Septic shock, shock caused by endotoxin of a gram-negative bacteria. Phagocytosis of bacteria leads to secretion of a tumor necrosis factor, which alters the permeability of the blood capillaries and causes them to lose large amounts of fluid. Low blood pressure affects kidney, lung, and gastrointestinal tract.